Sequencing genomes, the footprints of biological evolution. Determining the nucleotide sequences all the DNA in a genome is a formidable task. During the 1980s and 1990s, the technology to accomplish this effort gradually improved as researchers developed new vectors to clone large segments of DNA and increasingly automated procedures to determine the nucleotide sequence of these large fragments. The first complete sequence of prokaryotic organism was reported in 1995, and the first complete sequence of a eukaryote, the budding yeast, Saccharomyces cerevisiae, was published the following year. Over the next few years, as the scientific community awaited the result of work on the human genome, the genomic sequences of numerous prokaryotic and eukaryotic organisms, including a fruit fly, nematode, and flowering plant, were reported. Researchers were able to sequence these genomes with relative speed because they are considerably smaller than the human genome, which contains approximately 3.2 billion base pairs. To put this number into perspective, if each base pair in the DNA were equivalent to a single letter on this page, the information contained in the human genome would produce a book approximately 1 million pages long. By 2001, the rough draft of the molecular sequences of the human genome had been published. The sequence was described as rough because each segment was sequenced at average of about 4 times, which is not enough to ensure complete accuracy, and many regions that proved difficult to sequence were excluded. The first attempt to annotate the human genomic sequence, that is, to interpret the sequence in terms of the number of types of genes is encoded, led to a striking observation concerning gene number. Researchers concluded that the human genome probably contained in the neighborhood of 13,000 protein coding genes. Up till it had been sequenced, it had been widely assumed that the human genome contained at least 50,000 and maybe as many as 150,000 different genes. The finished version of the human genome sequence was reported in 2004, which meant each site had been sequenced 7 to 10 times to ensure a very high degree of accuracy, at least 99.99%, and the sequence contained a minimal number of gaps. The gaps that persisted contain regions of the chromosome often referred to as dark matter that consist largely of long stretches of highly repetitive DNA, especially those in and around the centromeres of each chromosome. Despite exhaustive efforts, these regions have proven possible to clone, or their sequences have proven impossible to order correctly using current knowledge or technology. Despite this remarkable achievement in nucleotide sequencing, the actual number of protein coding genes in the human genome remains uncertain. Identification of the genes using various computer software programs or algorithms has been plugged with difficulties, and in fact, the earlier estimate of 13,000 protein coding human genes has been revised steadily downward. Although it has come as a shock to most biologists, current estimates place the number in the neighborhood of 21,000. This means that humans have roughly and the same number of protein coding genes as a microscopic worm whose entire body, nervous system, and all consist of approximately 1,000 cells. It is evident from this idea that it is not possible, as was once thought, to understand the nature of an organism by simply knowing a list of the genes that make up that organism's genome. If the differences in complexity between the organism cannot be explained by the number of protein coding genes in their genomes, how can they be explained? We really don't have a very good answer to this question, but we can list a few possibilities to consider. First, as we described, a single gene can encode a number of related proteins as a result of a process termed alternative splicing. Several recent studies suggest that over 90% of human genes might engage in alternative splicing, so that the actual number of proteins encoded by the human genome is at least several times greater than the number of genes it contains. It is likely that greater differences between organisms will emerge when this and other gene enhancing mechanisms are explored further. This expectation is consistent with the observation that vertebrate genes tend to be more complex, contain more exons than those of flies and worms and exhibit a higher incidence of alternative splicing. So, molecular biologists have devoted an enormous effort to studying the mechanism that regulates gene expression. Despite this effort, our understanding of these mechanisms is very limited. We have learned, for example, that a large portion of the genome is transcribed into a bewildering array of RNA, but we have very little information as to what most of these RNAs are doing within the cell. There is a growing belief that many of these RNAs have a gene regulatory function. There is also growing evidence that the number and complexity of these non-coding RNAs can be correlated with the levels of complexity of diverse organisms. In one study, for example, researchers sought to identify the number of microarrays produced by various organisms. As discussed in the next chapter, microRNAs are one of the best studied regulatory RNAs. Researchers found that sponges expressed about 10 different microRNAs and CN amounts approximately 40. This compares to approximately 150 identified in worms and fruit flies and at least 1000 in humans. This is not to imply that the number of microRNA is the primary determinant of morphological complexity. Rather, it suggests that we will have to learn a great deal more about gene regulation if we are going to understand the basis of biological diversity. Over the past decade or so, a new area of biological study called systems biology has emerged that focuses on the ways that proteins work together as complex networks rather than as individual actors. A very simple example of a protein network was very important given that cells produce thousands of different proteins. When varying degrees of interaction, these networks can become very complex and dynamic. A relatively small increase in the number of elements that make up a network or an increase in the size and complexity of these elements could dramatically increase the complexity of the network and thus the complexity of the entire organism. Numerous other factors could be added to this discussion, but the general point is clear. The apparent difference in complexity between different groups of multicellular organisms is less a matter of the amount of genetic information in an organism's genome than that of the manner in which that information is put to use. Comparative genomics, if it's conserved, it must be important. Consider the following facts. The majority of the genome consists of DNA that resides between the genes, and this represents intergenic DNA. And each of these roughly 21,000 or so protein coding human genes consists largely of non coding proteins portions, intronic DNA. Taken together, these facts indicate that the protein coding portion of the genome represents a remarkably small percentage of the total DNA, estimated at about 1.5%. Most of the intergenic and intronic DNA of the genome does not contribute to the survival and reproductive capability of an individual, so there is no selective pressure to maintain this sequence unchanged. As a result, most intergenic and intronic sequences tend to change rapidly as organisms evolve. In other words, the sequences tend not to be conserved. In contrast, those segments of the genome that encode protein sequences or contain regulatory sequences that control gene expression are subject to natural selection. Natural selection tends to eliminate individuals whose genome contain mutations in these functional elements. As a result, these sequences tend to be conserved. It follows from these elements that the best way to identify functional sequences is to compare the genomes of different types of organisms. Despite the fact that humans and mice have not shared a common ancestor for about 75 million years, the two species share similar genes which tend to be clustered in a remarkably similar manner. For example, the number and order of human globin genes depicted is basically similar to that found in the complete segment of DNA in the mouse genome. As a result, it becomes possible to align corresponding regions of mouse and human genome. Human chromosome number 12, for example, is composed of a series of segments in which each segment corresponds roughly to a block of DNA from a mouse chromosome. The number of the mouse chromosomes containing each block is indicated. Even a quick examination of this drawing of a human chromosome, like 5, 10, 15, and 6 regions are shown. Dramatic change in chromosome structure that occur as evolution generates new species over them. Blocks of genes that are present on the same chromosome in one species can become separated into different chromosomes in a subsequent species. Over time, chromosome numbers can increase or decrease as chromosomes split apart or fuse together. One estimate suggests that approximately 180 breakage and fusion events have taken place in the mouse and human lineage since the time these two present day mammals last shared a common ancestor. Taken by themselves, changes in the positions of genes have very little effect on the phenotypes of organisms, but they provide a clear visual footprint of the evolutionary process. When homologous segments of the mouse and human genome are aligned on the basis of their nucleotide sequence, it is found that approximately 5% of DNA sequences are highly conserved between two species. This is considerably higher percentage than would have been expected by combining the known protein coding regions and gene regulatory regions, together accounting for roughly 2% of the genome. If we adhere to one of the foremost principles of molecular evolution, if it is conserved, it must be important. Then the studies are telling us that parts of genome that are presumed to consist of useless sequences actually have important unidentified functions. Some of those regions undoubtedly encode RNAs that have various regulatory functions. Others likely have chromosomal functions other than genetic functions. For example, these conserved sequences could be important in chromosome pairing, very important in chromosome pairing prior to cell division. Whatever the function, the genomic elements are often located at great distances from the closest gene and include some of the most conserved sequences ever discovered, exhibiting virtual identification between human, rat, and mouse genome. By comparing stretches of the genome between two distinctly related species such as human and mouse, one can identify those regions that have been conserved for tens of millions of years. However, this approach won't identify functional sequences in the human genome that have a more recent evolutionary origin. This could include, for example, genes that are present in humans and absent from mice, or regulatory regions that have changed their sequence over the course of primate evolution to allow them to bind new regulatory proteins. The concerted effect, called the ENCODE project, is underway to identify all of the functional elements present in the human genome. Unfortunately, we lack the knowledge necessary to recognize many of these functional elements, which obviously confounds the task. One point has become clear that recent studies, a significant proportion of functional DNA sequences are constantly evolving and thus are not highly conserved. In other words, if we restrict our search to conserved sequences, we run the risk of missing many of the most important functional elements in the genome. Next, the genetic basis of being human. By focusing on conserved sequences, we tend to learn about characteristics that we share with other organisms. If we want a better understanding of our own unique biological evolution, we have to look more closely at those parts of genome that distinguish us from other organisms. Chimpanzees are our closest living relative, having shared a common ancestor as recently as 5 to 7 million years ago. It was thought that a detailed analysis of the differences in DNA sequences and gene organization between chimpanzees and humans might tell us a great deal about the genetic basis for recently evolved features that make us uniquely human, such as our upright walk and advanced use of tools and language. These later characteristics can be traced to our brain, which has a volume of about 1300 centimeter cube, nearly 4 times that of a chimpanzee. The rough draft of the chimpanzee genome was reported in 2005. Overall, the chimpanzee and human genome differ by roughly 4%, which amounts to tens of millions of differences, a level of difference or divergence that is considerably greater than expected from preliminary studies. While some of this divergence is due to single nucleotide changes between the new genomes, most is attributed to larger differences such as deletions and segmental duplications. Researchers have been able to identify hundreds of genes in the human lineage that are evolving more quickly than the background, presumably, presumably in response to natural selection. However, it remains unclear which, if any, of these genes contribute to making us human. Some of the fascinating, fastest evolving genes encode proteins involved in regulation of gene expression, like transcription factors. These are precisely the types of genes that would be expected to generate major phenotypic differences because they can affect the expression of a large number of other genes. In fact, differences between chimp and human transcription factors are presumably responsible for many of the differences in expression of brain proteins. A comparison of the FOXP2 protein between humans and chimps shows two amino acid differences that have appeared in the human lineage since the time of separation from our last common ancestor. To assess the effects of these amino acid substitutions of FOXP2 function, human neuronal cells that lacked their own FOXP2 genes were engineered to express either the chimp or human version of the gene. These two cell populations were then studied in culture to assess the effects of the alternate versions of the transcription factor. It was found that more than 100 target genes were significantly upregulated or downregulated in cells expressing the human FOXP2 gene when compared to cells expressing the chimp version of the transcription factor. What makes the FOXP2 gene so interesting is that persons with mutations in the gene suffer from a severe speech and language disorder. Among other facets, persons with this disorder are unable to perform the fine molecular movements of the lips and tongue and required to engage in vocal communication. Calculations suggested that the changes in speech gene that distinguish it from the chimp person become fixed in the human genome within the past 1000 sorry about 200,000 years, a time when modern humans are thought to have emerged. An alternation in DNA sequence is said to be fixed if it is now present in virtually all membranes of the species. This finding suggests that changes in the FOX2P2 gene may be played an important role in the evolution of development of human speech. Many other genomic function differences between chimps and humans have also been described, including alteration in certain RNA encoding genes that appear to affect brain development. One of the genes in the later category, called HAR1, stretches for a mere 118 base pairs but contains 18 base substitutions that distinguish it from the corresponding region in the chimp genome. This same region is highly conserved among the other vertebrates, so it is only during the evolution of humans that it has experienced such pronounced effects of natural selection. HAR stands for Human Accelerated Region. Although the function of HAR1 is unknown, the fact that it is expressed primarily in the developing fetal brain makes it a gene of interest to researchers trying to unravel the genomic basis of human brain expansion. Another gene of interest encodes the starch digesting enzyme amylase that is present in saliva. Chimps have a relatively low starch diet and have one copy of AMY1 gene in their genome. During the evolution of humans, there has been an apparent selection for an increased number of copies of AMY1 gene, which has results in an increased concentration of the amylase enzyme in human saliva. One might have predicted that an increased amylase level would have been accomplished during evolution by an increase in the level of expression of a single existing AMY1 gene in the genome, but in its in this particular case, it has resulted from gene duplication. The number of copies of the AMY1 gene among humans is variable. In fact, the number of copies of the gene tend to be higher in human population that ingest higher quantities of starch in their diet. One of the most interesting questions in the area of human evolution concerns possible relation between modern humans or Homo sapiens and those of other human species like archaic members of Homo genus that were now extinct, humans that would be distinguishable from our present species, that is, anatomically modern humans, are thought to have first evolved in Africa approximately 200,000 years ago. But Homo sapiens are not the only members of the Homo genus to have inhabited Earth in the past 200,000 years. In fact, another member of the genus Neanderthalensis, Homo neanderthalensis, was a resident of the Europe as recently as 35,000 years ago. Thousands of years after the arrival of modern humans on the continent, the two species are thought to have shared some of the same regions of the Middle East at an even earlier time period. Given that both of our species inhabited similar areas and were very similar anatomically, paleontologists have wondered whether the two species had interbred or modern humans had simply replaced the Netherlands without Neanderthalans without interbreeding. Neanderthalans without interbreeding. An answer to that question requires detailed information about the differences in DNA sequences between modern humans and Neanderthalans. Beginning in the late 1990s, researchers have developed increasingly sophisticated technique to isolate and sequence snips of mitochondrial DNA, mtDNA, from fossil remains of Netherlands. Mitochondrial DNA is easier to analyze from fossils than is nuclear DNA because of each cell contains many copies of the mitochondrial genome and it is much smaller in size. Results of the study suggest that Neanderthal mtDNA sequences are sufficiently different from that of modern human mitochondrial DNA to conclude that Neanderthals went extinct without contributing genetic material to the modern human mitochondrial genome. In other words, the two species had no interbred and, moreover, had not shared a common ancestor for at least 300,000 years. By 2010, Savant Pabo and colleagues at the Max Planck Institute in Germany had assembled a relatively complete sequence of the Neanderthal nuclear genome and concluded that modern humans and Neanderthal genomes are 99.84% identical. Neanderthals, for example, had the same POXP2 allele as do modern humans, which hinted that they too had verbal language skills. Moreover, the detailed comparison of the molecular genomes of a number of different Neanderthal fossils and those of several present-day humans suggests that approximately 1 to 4% of DNA of modern Europeans and Asians is derived from a Neanderthal source. Many of the genes in the Neanderthal-derived DNA contribute to the immune system recognition by pathogens. Acquisition of these genes may have served to protect the newly acquired humans from disease to which the Neanderthal had previously been exposed. In contrast, the genomes of Africans show no evidence of a contribution by Neanderthals. According to this data, modern humans and Neanderthals interbred at some point in time and space after modern humans had left Africa and before they had spread into Europe and Asia. Other reports suggest that some groups of modern humans have ancient ancestors that may have interbred with other archaic human species besides Neanderthals. This finding have set the stage for some very interesting speculation concerning the human phylogenetic tree. According to some researchers who envisioned the number of human species sharing the planet over the past 100,000 years or so, our present population can be described as the last humans standing. We have focused on the section on genome differences between humans and other primates because this is the subject of the present chapter. It is probably evident from the limited nature of this discussion that researchers have not yet made much progress in identifying the genetic basis of being human. Many researchers believe that too much attention in this field has been focused on changes in protein coding sequences. Instead, they would argue that changes in the regulation of gene expression have played a primary role in human evolution, but it is difficult to ascertain which of these changes have real importance. Genetic variation within the human species population. No one else in the world looks exactly like you because no one else, other than an identical twin, has the same DNA sequence throughout their genome. The human genome that was sequenced by the original Human Genome Project was derived largely from a single human male. Since the completion of that sequence, a great deal of attention has been focused on how DNA sequence varies within the human population. Genetic polymorphisms are sites in the genome that vary among different individuals. The term usually refers to a genetic variation that occurs in at least 1% of the species population. The concept of genetic polymorphisms began with the diversity discovery in 19th century by the Australian physician Carl Landsteiner that people could have at least three alternate types of blood ABO. ABO blood grouping is a result of members of population having different alleles of a gene according to a sugar transferring enzyme. Now that we are in possession of our own genetic sequence, we are in a position to search for new types of genetic variation that would never have been revealed in the pre-genomic era. Then DNA sequence variation, the most common type of genetic variability in humans, occurs at sites in the genomes where single nucleotide differences are found among different members of the population. When present in at least 1% of the population, the sites are called single nucleotide polymorphisms. The vast majority of SNPs, pronounced SNPs, occur as two alternate alleles such as A or G. Most SNPs are thought to have arisen by mutation only once during the course of human evolution and are shared by individuals through common ancestor. Commonly occurring SNPs have been identified by comparing DNA sequences from the genomes of hundreds of different individuals from diverse ethnic populations. On average, two randomly selected human genomes have about 3 million single nucleotide differences between them, or 1 every 1,000 base pairs. Although this might appear to be a large number, it means that humans are, on average, 99.9% similar to one another with respect to nucleotide sequence, which is probably much greater than most mammalian species. This sequence similarity reflects the fact that we are apparently a young species in evolutionary terms and our prehistoric population size was relatively small. Of the 15 million or so SNPs in the genome, it is estimated that approximately 20,000 has found in within protein coding sequences and led to amino acid replacements within the encoded protein. This corresponds to about one amino acid substitution per gene. It is this body of genetic variation that is thought to be largely responsible for the phenotypic variability observed among humans. The impact of human genome sequence variation on the practice of medicine is very important. Then, structural variation. Segments of the genome can change as the result of duplications, deletions, insertions, inversions. This type of changes typically involve segments of DNA changes from hundreds to millions of base pairs in length. Segments of the genome can change as the result of duplications, deletions, insertions, inversions when a piece of DNA exists in a reversed orientation and other events. This type of change typically involves segments of DNA ranging from hundreds to millions of base pairs in length. Because of this relatively large size, this type of polymorphisms are called structural variants, and we are just beginning to grasp the extent and importance of their presence. It has been known since the early days of microscopic chromosomal analysis that not all healthy people have identical chromosome structures. Chromosome inversion, whose existence has been known for over 30 years. Recent studies have revealed that intermediate sized structural variants, those that are too small to be seen under the microscope and too large to be readily detected by conventional sequence analysis, are much more common than previously thought. According to an estimate, a typical human genome carries approximately 1,000 structural variants ranging in length from about 500 bases to 1.3 million bases. Although the estimates vary considerably, it is evident that the fraction of the genome total number of base pairs affected by structural variation is greater than that affected by SNPs. As a result, structural variation has a significant effect on phenotypic variation among humans. Copy number variation is a technique of DNA fingerprinting depends on the differences in the lengths of mini satellite sequences, which in turn depend on the number of copies of the sequences that are present at particular sites in the chromosome. This is an example of copy number variation or CNV. Recent findings have revealed that much larger sized CNVs are also common in the human population, affecting approximately 10 to 15% of the genome, including large number of protein coding genes. This large sized CNVs are a type of structural variation that results from a duplication or deletion of a DNA segment. Because of such CNVs, many of us carry extra copies of one or more genes that encode important physiological proteins. Extra copies of genes are generally associated with overproduction of a protein, which can disrupt the delicate biochemical balance that exists within a cell. It is found, for example, that a significant number of persons who develop early onset of Alzheimer's disease possess extra copies of the APP gene, which encode the protein thought to be responsible for the disease. Although the subject has been controversial, there is a growing consensus among researchers that individuals with autism or schizophrenia have a greater than normal number of rare CNVs in their genomes. Some of the genes affected by the CNVs is involved in synapse formation and other neurological process. In some cases, extra copies of genes can be beneficial, as illustrated by the repeated duplication of the amylase gene AMI1, which has been found in certain human populations that have a high starch content in their diet. This is an excellent example of CNV in that it shows the number of AMY genes on both copies of one person's homologous chromosomes. It can be seen that one chromosome contains only 4 copies of AMY1 gene, where the homologous chromosomes contain 10 copies of the gene.